All right. It is my great pleasure today to introduce Drs. Barry Everitt and Trevor Robbins from University of Cambridge, who will be telling us a little bit about their influential thinking and paper in response in psychopharmacology to Dr. Kent Berridge's summary of his thinking at the time on dopamine in motivation and other reward-related functions. And I will now hand it over to Drs. Everitt and Robbins, who will do kind of a tag team type presentation here on their thoughts on this important topic. Drs. Everitt, Robbins, take it away.
Okay, thanks very much, Steve. And thank you for inviting us to your, what can we call it? Your chat show.
I guess so. It's Psychopharm Friday is what we're doing here.
Psychopharm Friday.
For the journal Psychopharmacology.
Got it. I think probably what I should do, and Trevor and I are we've got some pieces that we'll say individually, but begin by saying that we very much enjoyed Kent's sabbatical stay in Cambridge, those many years ago now, that he described in his interview. And we were thrilled to welcome him as a distinguished visiting scientist to the Department of Psychology and to Downing College. And the seminars, of which there were many that we held there, were memorable, but I would say more for their bonhomie and erudition, rather than being a battleground for the dopamine wars, as they've been alluded to in some of the text that's been flying around on this. I don't remember any wars, but Trevor, do you?
No, I don't either. We were very pleased to be able to write this commentary on this highly influential and highly cited review that Kent did for Psychopharmacology. And I think it's fair to say that many of the points we raised at the time in that commentary, for us, still hold today. Maybe there are some new things that have been raised since, but a lot of our original arguments, I think, still hold true. And quite a lot of our points agreed, actually, ultimately, with Kent's position, particularly regarding incentive sensitization. Although I should say that, we'd shown quite a lot earlier than he talked about incentive sensitization that amphetamine-like drugs enhance reward-related stimuli, which is what I assume incentive sensitization is, and that this depended on striatal dopamine. That was work done by Jane Taylor in our lab. So we accorded with that a good deal, except we called it what it was, which was an enhancement of the effects of conditioned reinforcement, which is a trifle more technical sounding than incentive sensitization. And nevertheless, we still found, of course, that this effect of enhanced conditioned reinforcement played an important role in animal models of cocaine drug seeking, as in our work on second order schedules. So in other words, the drug seeking was held together by these drug paired stimuli, which the animal would work for in advance of its intravenous cocaine self-administration. So I think that's quite an important point to make, and it's something we're returned to. Now, of course, we have emphasized this role of instrumental actions in self-administration of cocaine and in drug addiction, and also the transition of instrumental actions to habits. But these behaviors are mediated by different segments of the stratum, indeed the dorsal stratum, rather than the nucleus accumbens and ventral stratum. And it's clear that dopamine probably modulates all of these areas to produce somewhat different functional effects. So we don't think that dopamine is simply involved in incentive sensitization, but also, for example, in motor readiness and the ability to initiate actions. I mean, after all, Parkinson's disease is not a disease of incentive sensitization. It's a problem of movement initiation, and it's crucially dependent, as we all know, on depletion of dopamine from the dorsal stratum. Now, it's true that Parkinson's disease also has cognitive impairments and also some motivational problems, which can be exacerbated by overdosing from D2 agonists. And this arises from this imbalance in dopamine depletion across the stratum. But it's clear that dopamine is generally affecting functions in these different areas in different ways, in the domains of motor action, cognition, and motivation. And of course, originally in the 90s, actually, just referring back to John Salamoni's podcast, we were talking about dopamine and its role in activation, a rather general process incorporating motivation, but also taking into account effects on novelty seeking and also on stress and aversive responses. Many of dopamine's effects aren't simply to do with positive incentive processes. They may also infect aversive processes as well. So I guess this is, these are very important points which we'll come back to. And of course, in the 2008 commentary we did, we also took on board some of the new work by Wolfram Schultz on prediction errors and dopamine and learning. And we were forced essentially to modify our own theory on that basis. And I think Barry's going to take up this particular element.
Yeah, okay, I will. And I think what I say there is, unlike Kent, we don't find it difficult to embrace the data of Wolfram Schultz and others that phasic dopamine neuron firing might be implicated in the construction of prediction errors that contribute to reinforcement learning. And although many effects of drugs affecting dopaminergic systems probably depend on their effects on tonic dopamine function, it doesn't really surprise us that the system is also endowed with plasticity to enable new learning and memory. And in our commentary back then, was it really 2008? I realize that is a long time ago. We pointed out what was then a recent study by Jeff Daly here in Cambridge showing post-session effects of intra-accumbens dopamine receptor agents on the consolidation of stimulus-reward associations. And I think it's also important to mention that Norman White and his colleagues, around the same time, a little bit earlier it may have been, showed analogous effects on the consolidation of aversive memories following post-session dorsal striatal manipulations. So overall, on the basis of all these results, it appears a little naive to us to believe simply and monolithically that dopamine is incentive salience. I mean, the other thing I go on to say is that instrumental behavior doesn't feature greatly in Kent's writing, yet appetitive behavior, which has been key to motivational theories since Bindra and Konorski, that are much loved by Kent and by me and by Trevor, I'm sure, involves instrumental seeking or foraging behavior that gives an animal access to rewards having wanted them, or when they're elicited by reward-related stimuli, especially conditioned reinforcers that are there in the environment. And if I could just refer as well to some recent work suggesting why it is we need to think beyond incentive salience and more generally and more encompassingly about dopamine. It's work that was initiated here by a grad student when Kent was here called Sitsi Yonkman, who went to Paul Kenny's lab, and they completed what was about a decade's worth of research. First author is Alexander Smith. Published additional responses to food reward. Inhibiting dopamine D2 receptor MSNs further strengthens that consolidation, but blocks the expression of previously learned habits. So this does seem difficult to interpret these data in terms of an incentive salience function of dopamine, unless the term has an all-encompassing, highly flexible, and therefore not very informative meaning. And if I may just go on for a little bit longer, if that's okay with you and Trevor, we do agree with Kent that Roy Wise's bold idea that dopamine systems mediate pleasure may be incorrect. And Kent has, of course, in his superb APA award-winning earlier work, provided evidence against that view by showing that manipulations of striatal dopamine fail to influence reflexive ingestive responses that appear to reflect approach or liking or aversive reactions. And Trevor just alluded to John Salamone, and he and others have also provided evidence against a simple hedonic interpretation of the effects of dopamine manipulations. However, equally, it's difficult, I think, to infer what are subjective responses of humans from the behavior of other animals. And such anthropomorphic descriptions of behavior can lead to unfortunate mistakes. So we're therefore unsure that the rejection of liking in favor of a motivational construct of wanting adds much to the explanation of the role of dopamine, certainly in the context of drug addiction, where aversive responses to withdrawal from certain drugs also play an important role. And frankly, it's never really been clear to me anyway, how reflexive orofacial responses to food taste in rats that indicate liking translates to a self-administered stimulant like cocaine. I mean, what is a cocaine liking response in rats? Could it be a lever press? But that's the measure of wanting. And wanting itself connotes a goal and behavior directed to it, which is known to be mediated by dorsal medium striatal circuitry, as Trevor indicated later, rather than the nucleus accumbens, which mainly mediates Pavlovian associations and their transfer to instrumental behavior. So without an instrumental response, an animal might indeed approach stimuli associated with food or intravenous cocaine, but they wouldn't be able to gain access to these reinforcing events. And it's always seemed to us that a simple Pavlovian function of mesolimbic dopamine, whatever you may call it, isn't really adequate to explain the transfer to action that's required to forage for and consume a reward, whether it's a drug or food. But I'm sure, Trevor, you have more to say about this.
Yeah, well, it does come back, Barry, doesn't it, to this difference between a Pavlovian conditioned stimulus and a conditioned reinforcer. And obviously, a conditioned reinforcer evokes action as a consequence of the behavior. And so we think that's a really important link in addiction. So, for example, we don't find very strong effects, in my estimation, for Pavlovian to instrumental transfer in maintaining or enhancing self-administration behaviors for cocaine. It's only when the stimuli are contingent on the behavior as conditioned reinforcers that you get that exaggeration and control of behavior for cocaine, for example, which is presumably dopamine mediated. And in general, just trying to draw things together, a theory of mesencephalic midbrain dopamine is likely to be quite complicated. It depends on a number of different theories, really different hypotheses, and a compromise has to be reached among them because they all have a point to make. We have to consider, for example, the role of dopamine in different domains of the stratum and also the prefrontal cortex and the amygdala and the hippocampus even. We also have to consider the differences between phasic and tonic modes of dopamine action. And as you've already made the point, phasic modes are particularly important for plasticity and learning, which are superimposed on this tonic mode. Additionally, we have different dopamine receptors. You've alluded to D1 and D2 receptors, which probably have different functions. And of course, these are D1, D2 light receptor families. And also the whole problem of the difference between learning and performance and memory, and how motivation can't possibly be an all-embracing construct which accounts for all of these things. So I think that, you know, there are different components of these positions which need to come together in a better integrated theory about dopamine. And that was the gist of our original commentary. And it's also been the gist of our work on addiction, where, for example, we don't ignore the role, for example, of drug sensitization in addiction. It may be an important component, but we also stress this role of habit learning and other factors in addiction, top-down cognitive control, for example, to produce a more integrated theory, rather than suggesting that, for example, addiction is a consequence of drug sensitization. So in general, although we think a hypothesis testing, parsimonious approach to science is important, and you have to reject hypotheses, you can't reject whole bodies of data or theories just on, on the account that another position seems to work. I don't think things are as simple as that. A very famous dopamine neuroscientist, to paraphrase, has said something like, there's more to incentive sensitization than dopamine, and there's more to dopamine than incentive sensitization. And I think that's very much our kind of position here. And although we think that Kent's a fantastic scientist and has promulgated this theory with great verve and thoughtful insistence, we think the notion that dopamine is incentive sensitization is just a little bit too narrow.
Great. I have a question regarding that. So would you guys say that the idea that there is a function for dopamine is a plausible scenario? Because you did lay out some important points that the sort of receiving end of this dopamine signal or signals, whatever it is very important here. For example, a key example would be the D1, D2 in striatum. These neurons are going to do opposite things in terms of their activity, presumably in response to dopamine. So is it plausible that there is sort of a function of dopamine and that all the heterogeneity you refer to is coming from the target regions, so to speak, how it interprets that signal? Or would you lean more on the side of there really is a functional gradient between the medial and lateral populations of dopamine neurons in the midbrain, for example?
Well, my position on this, Barry may have a different view, but my position on this is that it's going to be difficult to say that that function of dopamine is a psychological construct. It's more likely to be something very abstract and physical about, the way dopamine changes signal to noise ratios in particular regions. And that's the kind of theory which I think is ultimately going to provide a more general account of what dopamine and similar neuromodulators do. The point about dopamine's functions depending on its terminal domains is very important, I think, because clearly if dopamine is modulating processing in the hippocampus, it's very unlikely that those functions are going to be behaviorally similar to those in the putamen, for example. One would have thought. That's perhaps not the way to think about a general function for dopamine. So it may be a semantic issue, but that would be my position.
Yeah, I concur. And somehow we've never had that difficulty in conceptualizing, for example, the functions of cortically projecting noradrenergic neurons. In such, we don't say noradrenaline is something like incentive salience, but as applied to noradrenaline, we can talk more generally in terms of what it does on the processing and neuromodulation in terminal regions, such as, for example, to enhance signal to noise ratio, which it does to touch in the somatosensory cortex and to visual stimuli in the visual cortex. It's a kind of a general function that is utilized according to the processing that's going on in the areas that it innervates. And I think this probably reflects a different way that Kent and we have approached function, and we've always been interested in dopamine function, but we've very much more been interested in thinking about it in terms of corticostriatal systems and the modulation of their functions, whether in behavior output or learning and indeed memory, which we determine experimentally, as a setting in which to study them.
Yeah, for example, think about serotonin. Try and come up with a unitary function for serotonin. It's a nightmare. And the other point I would make is that I guess Barry's and my research since 2008 have been less dopaminocentric, perhaps, than the rest. I mean, we're very much more interested in the overall networks, the corticostriatal networks, for example, that govern cognitive control over these behaviors, whether they're habits or instrumental actions or Pavlovian responses. And of course, dopamine plays an important modulatory role in all that, but it's only a small part of a bigger picture, which we're going to need to have to understand addiction, OCD, schizophrenia, and so on. And Parkinson's disease, by the way.
Mm-hmm.
So yeah, certainly I think these are fantastic points. I wonder though, this sort of gain modulation or signal to noise ratio type functions of, you mentioned some of multiple neurotransmitters, these sort of midbrain or hindbrain modulators. This is sort of a general function that one could, say similar things about norepinephrine and dopamine, as you just did. Would you say that it's fair to assume that there is some difference qualitatively between dopamine and norepinephrine, let's say?
Yeah, well, we've actually, Amy Arnston and I discussed this in our paper in Annual Review a few years ago. And yeah, the evidence that Amy's mainly responsible for has been to show, for example, that D1 receptors and alpha-2 receptors in the prefrontal cortex affect signal-to-noise ratio in different ways. One of them boosts the signal and the other one reduces the noise, for example. So I can imagine all sorts of those kind of syntactic functions of these transmitters are working together to process the signals in these terminal areas. And we really just don't have the vocabulary yet to understand what we could label this as. I mean, Barry and my position, if you read some of our other papers, has always been that these systems are all kind of differentiations of the old arousal system. And what we're all talking about, really, I think, with these systems, serotonin, dopamine, noradrenaline, histamine, the whole rest of them, is they're different chemical elements of the old reticular core, and they're contributing different types of modulation on terminal processing. They're probably all subject to rather different predisposing influences, which we call stress, which is another big monolithic term, which probably has to be fractionated. And that's the subtlety of it. I mean, these transmitters are all contributing something subtly different to cortical or forebrain processing. I think that's our general view. And dopamine is no different from the others in that regard.
In that regard, yeah.
But it is fascinating, isn't it? For example, John, that dopamine innervates the entire stratum, noradrenaline just innervates a bit of the shell. That's kind of interesting. Those are the kind of things that interest us. Why is it like that, for example, And, the dopamine innervation of the hippocampus has been pretty marginal, although recently, as you probably know, Richard Morris and others have discovered this role of D1 receptors there, although the projection to the hippocampus is pretty minor. So there's a lot of things we don't understand yet about dopamine.
Sure. As I understand it, that story involves norepinephrine neurons using dopamine transmitter. Right?
Yeah, exactly.
Exactly.
So that's when it gets a bit complicated. Yeah,
sure.
It's very sneaky.
Wow. So beyond that particular finding, are there any other ways in which your view of dopamine or your position on these topics has changed since 2007 that you'd care to talk about?
Well, as I said, a lot of the points I think we raised are still valid by and large. I don't think many of them are wrong. Obviously, there's been a lot of very sophisticated computational work since then. I think the jury's still out on how useful that's going to be ultimately, but I think that's been an interesting approach. And obviously, the optogenetics approach has been quite interesting. But again, I think perhaps Barry would like to comment on this. I think the jury's still out a little bit on what we've learned new about dopamine with optogenetics.
Yeah, that sounds like descending into the heretical abyss, doesn't it? But I mean, I can think of sort of quite simple ways in which our views have had to change. For example, when Trevor and I first started working together, which is before
200 years ago.
Yeah, before 2008. One of the first things we in fact, Trevor showed was that if you took dopamine away from the striatum, particularly the ventral striatum, it didn't stop animals undergoing Pavlovian conditioning in a conditioned reinforcement paradigm. So stimuli, which are having incentive salience attributed to them, can do that in the absence of a dopamine system in the nucleus accumbens, either because it's been knocked out with a 6-OHDA lesion or dopamine receptor blockade in accumbens. And what stopped the acquisition of a new response with conditioned reinforcement Okay, so this translation of Pavlovian conditioning to action was interference with the amygdala. And we showed that the amygdala and nucleus accumbens interact as a corticostriatal system that imbues Pavlovian conditioned stimuli with the power to act as condition reinforcers. And the dopamine innervation of the accumbens, particularly the shell, massively amplifies that control over behavior by condition reinforcement. So early on, it seemed to us that dopamine wasn't required for, so Kent would have loved this, right? Dopamine isn't learning, it's not required for learning. And that's kind of what we saw. And I remember us having these debates with Wolfram Schultz when he came to Cambridge. You need to do an intervention, Wolfram, to show that something's causally involved in a process rather than just going for correlation. But I think the advent of prediction error and reinforcement learning and these effects of tonic, post-session, it can't be an incentive motivation effect. Post-session manipulations after a conditioning event help consolidate and strengthen the Pavlovian relationship. We've had to change, a view based on that. But so frankly, I'm a little bit puzzled that Kent so strongly opposes anything to do, well, there's this prediction error stuff, yes, but learning, it's not really learning. So I think this is, again, this approach that Kent brings to his work, which is great, makes it great fun to interact with him. But this is my view, these are the experiments I've done, this is what I found, that's wrong, that's wrong, that's wrong. When he combined with Terry Robinson in the famous 93 paper, which they sent me to comment on, actually. I was doing a sabbatical in California at the Scripps, and they sent me this to comment on, and they acknowledged me in that as well. It was a strong position that was so strong that it said, well, forget about withdrawal. Withdrawal is absolutely nothing to do with addiction. It is this positive incentive, increase in wanting in the face of decreased liking, that explains addiction. I mean, I love the provocative nature of that because it makes everybody experiment on it. Just like Roy Wise's hypothesis did, dopamine's pleasure. The world and his brother have jumped on that, but actually Roy fulfilled a great service by preventing such a strong statement that people could then, I'm not sure Roy ever changed his mind dramatically, but Kent never seems to. So it's very interesting. And for example, this notion of incentive salience sensitization, we've always acknowledged it is probably a very important bottom-up contributor to compulsion. So is top-down removal of cognitive control, as is the aversive state of withdrawal. And I'm always taken by Rajita Sinha's work on craving, which doesn't sound like a very positive dopamine incentive salience process. It sounds much more like a Kubian negative reinforcement aversive process. So there, this use of words, craving, for example, wanting, liking, it's useful, it's a tag to hang on, but sometimes I think it can confuse when we're dealing with studies of animal behavior.
I think the other thing you asked me about the new findings which make us change our views, but I do think a very interesting source of evidence is coming out from work by people like Joshua Burke and others about different, and Hikosaka's group, about different dopamine neurons even in the midbrain, which seem to have different properties. It's not just this type of prediction error, it's that type of prediction error, or it's not related at all to prediction error. I think there's more heterogeneity of dopamine cells in the midbrain than we certainly realized early on.
Even transmitter content, as we're
Indeed.
increasingly
and undoubtedly that. And of course, they all have different inputs as well. That's the other, which makes it even more complex. And as I was saying earlier, hard to make a generalization, probably.
And those top-down projections that regulate the activity of the brainstem monoamine neuronal groups, right? I mean, that's, I don't think dopamine neurons go out into the world and define what's salient or not.
Well, you know, even in the human imaging evidence suggests that some of the prediction errors come from the frontal cortex.
Yeah, absolutely.
You know.
Sounds like we have a lot of work to do here.
Well, I hope so.
Yeah. Anyway, it's been a very, very enjoyable discussion, as it continues to be. Yeah.
Absolutely, as has this been. Thank you both so much for your time. This has been really fascinating. I'm sure the rest of the scientific community watching this will agree, and hopefully this will generate a sort of next wave of discussion of these matters, I hope.
They might also scream and tear their hair out.
Well, that's true.
But maybe we could wish everyone a happy new year in any case.
Thank you both so much. Really appreciate your time.
Bye.
Good to talk to you, Steve.